Ken, this is my third time trying to tape this video, so this is all or nothing, alright, because my battery's about dead. Alright, this is lab 10, and on lab 10 we are constructing DNA to start with, and then we're going to see how RNA takes the message from DNA and takes it into the cytoplasm to make a protein where we get our traits. This is known as protein synthesis. Alright, so on your instructions it said you have a strand of DNA. One side of the DNA is these bases. These are nitrogen bases because they contain nitrogen. The G's and the A's, adenine and guanine, are twice as big as the C's and the T's because they're two carbon rings, where the cytosine and the thymine are only one carbon ring. How do we remember which is a purine, which are the big ones, which are pyrimidines? Because Georgia Bulldogs, G and A, love Purina dog chow, so they're the purines, the double rings. Alrighty. Now, the other side of the DNA is going to be the complementary bases to this. And so they would be these. If I can get them to turn right on my tape. Alright, so what would go with C? It would be a G. What would go with the A? It would be thymine, a T. We go with another C, it would be guanine. A goes with T, G goes with C, G goes with C, A goes with T, and C goes with G. Okay, so in DNA, we also know the sides of the ladder are made up of deoxyribose and phosphates. Where there's every base, there's a deoxyribose and phosphate. That subunit is called a nucleotide. What are these bonds that hold the rungs of the ladder together, hold these C's and G's together, the nitrogenous bases? Those are hydrogen bonds. Are they strong? No, they're pretty weak. So we also know that a segment of DNA that codes for a particular protein is known as a gene. And in this case, we're going to see how that gene is going to be copied and the code carried. So the first thing that will happen is there is an enzyme called helicase, the naughty enzyme that likes to unzip the genes. It comes in, cuts these hydrogen bonds, and peels away that one side of DNA. And then there's another enzyme called RNA polymerase. The RNA polymerase is going to start reading these bases and making the messenger RNA. So everywhere there is a C, it's going to bring a G. Everywhere there's an A, it's going to bring the complement of that. Is it going to be thymine? No, it's going to be uracil, because we learned in our learning objectives that RNA does not have any thymine. The uracil replaces it. So instead of any T's, it's going to be U's. And it's complementary base. It's with adenine. So everywhere there's an A, we put a U. Everywhere there's a C, we put a G. Everywhere there's a T and A, a T and A. Everywhere there's a G, we put a C. Everywhere there's an A, we put a G. A C goes with the G, A goes with the U, and a G goes with the C. So now we have just made our messenger RNA. This is known as transcription. Transcription happens in the nucleus where the DNA is. Now the messenger RNA goes out of the nucleus, out the nucleic pores, down into the cytoplasm where we're going to do translation. So translation occurs in the cytoplasm. What's it going to take to translate this messenger RNA? A ribosome, also known as ribosomal RNA, the second type of RNA. We know a ribosome is a big subunit and a little subunit, like a big rock and a little rock. This messenger RNA is going to be read by that ribosome. It's going to go down through there, and it's reading it 3 letters at a time. We know those 3 letters are codons. So it reads this codon, GUG. All right, I got the codon GUG. What is the amino acid that is coded by that base pair or base codon of GUG? We've got a table that tells us. First letter was G, second one was U, third base was G. So if we look in this box, GU, and then bring this one over G, we find GUG is right there, and it is coding for valine, the amino acid valine. So here comes our tiny transfer RNA. It comes flying in. It's got the anticodon of CAC, so that it goes with the GUG. It's the complementary pair of it. So it says, here I am, I'm dropping off my amino acid. It drops off its amino acid, valine. And the ribosome goes on and reads the next codon, which is AAC. So we've got to find out what amino acid goes with AAC. First base was A, second base was A, so we go down here under this A. Third base was C, we go down to C. AAC is right there, and it's coding for asparagine. And here comes the little t that's got the anticodon for that. UUG is the complement to AAC, and it drops off asparagine. And the ribosome is going to weld those two together with the peptide bonds. It's going to read the next codon, which is GUC. So after it reads GUC, we see the first letter was G, the second one was U, the third one was C. We find this box, GUC. It is another codon that goes with the amino acid valine. There can be more than one codon for amino acid. So now this little t comes flying in. It says, I got the anticodon for GUC. It is CAG, the complement to it. So it drops off another valine. And these amino acids are put together with really strong bonds, peptide bonds. That chain would just keep going longer and longer as this ribosome read more codons until eventually that chain would be a polypeptide chain and have thousands of amino acids that would curl up into proteins. So this is all happening in the cytoplasm. The DNA and the messenger RNA that's formed is in the nucleus. Then the messenger RNA goes out of the nucleus into the cytoplasm, where the ribosome reads it 3 letters at a time, codon by codon. The transfer RNA comes flying in and drops off its amino acid to be welded together by those peptide bonds. And that, my friends, is Lab 10 in a nutshell. See you later. Bye.